So this is the third of 4 videos regarding cell communication or cell signaling. And this one regards signaling transduction pathways. And by the time you're done watching this video and reading the text, I want you to be able to describe the difference between kinases and phosphatases, describe how kinases and phosphatases are involved in phosphorylation cascades, define second messengers, describe the role of calcium and cyclic AMP and IP3, these second messengers, in signal transduction pathways, and describe the function of the two enzymes adenylyl cyclase and phospholipase C. So here we have the overview of cell communication, where we have the signal binds to the receptor, which activates a signal transduction pathway leading to a cellular response. So we're going to focus here on what is this stuff going on that connects the signal to the cellular response. Now, much of a signal transduction pathway depends upon kinases. And kinases are enzymes that catalyze the transfer of a phosphate group from ATP onto a molecule. So in the situations we're going to look at, onto a protein. And now once a protein has become phosphorylated, that is, it has a phosphate group that's been covalently attached to it by a kinase from ATP, it now is either active or inactive. Now, phosphatases lop off these phosphate groups. They lop it off, and so now the protein has become dephosphorylated. What I want you to do is to take a moment and look at the activity of each of these enzymes, kinases and phosphatases, and see whether they are truly complete opposites of one another. Okay? I'm not going to give you the answer on that. We can talk about it in the lecture. Now, a phosphorylation cascade involves a series of kinases that start off inactive and then become phosphorylated by another kinase to become active. So let's take a look at what I mean by that. Here we have a receptor. Here we have the signaling molecule. It binds to the receptor, activating it. That active receptor turns on a molecule that activates our first kinase. So now that kinase is active, and therefore it phosphorylates our second kinase. Well, now that kinase is phosphorylated, it is active. And so then it phosphorylates our downstream enzyme, our final enzyme that we're interested in, and therefore activating it. And now that enzyme catalyzes a reaction necessary for the cellular response. So we get this cascade of phosphorylation from one kinase phosphorylating another, and then finally you activate that enzyme involved directly in the cellular response. And the question that you might ask is, why so indirect? Why not just connect this receptor to this protein right here and be done with it? Why all the in-between stuff? Well, there's two really important things that come out of this indirect pathway. The first is something called signal amplification, and we'll take a little bit more look at it a little bit later. But what that basically means is that each step of the pathway amplifies the signal. So one activated molecule will activate maybe 10 of these kinase 1s. And then maybe 10 of these kinase 1s will activate 100 of these kinase 2s, which will then activate 1,000 of these inactivated proteins. And so by that means, a single signal can actually activate many proteins down here, leading to a much more robust cellular response. The other reason that these signal transduction pathways have evolved to be so indirect is that they offer multiple control points of regulation. So that if there is something else that some other signal that cell is receiving that may want to override this signal, that you have all of these various control points, all of these proteins that can be inactivated by competing pathways. Okay, so it's a form of regulation. In addition to phosphorylation cascades, another common feature of signal transduction pathways are second messengers. And second messengers are defined as small, water-soluble, non-protein molecules involved in signal transduction. And we're going to look at several of them. The first is called cyclic AMP, or sometimes abbreviated CAMP. And cyclic AMP is made from a molecule of ATP. So a molecule of ATP is a substrate from the enzyme adenylyl cyclase. And what that does is it plucks off two of the phosphates and connects that third phosphate back on top of the molecule. So here is when we talk about the cyclic part of cyclic AMP, it's the fact that this phosphate group is reattached right back to that molecule. And cyclic AMP activates protein kinase A. So now if we take a look at a full kind of signaling response here, we've got our old friend, the G protein coupled receptor and a G protein. So what we have is this. Remember, each G protein coupled receptor needs three things. It needs the receptor, it needs the G protein, and it needs some other downstream enzyme attached to that membrane. In this case, it's adenylyl cyclase. So when the first messenger, our signaling molecule, binds to the receptor, it activates the G protein. The G protein activates the adenylyl cyclase. That catalyzes the production of cyclic AMP from ATP. That cyclic AMP is an allosteric activator of the protein kinase A, meaning it turns it on, and then we end up with maybe a phosphorylation cascade that eventually leads to a cellular response. So this is how phosphorylation cascades and second messengers aren't necessarily mutually exclusive. They all exist or can exist within the same signal transduction pathway. Calcium is another example of a second messenger. And it turns out that when you look at a cell, there are calcium pumps that work very specifically to reduce the concentration of calcium ions in the cytoplasm and increase their concentrations within the endoplasmic reticulum and the mitochondria. So you've got these calcium gradients that exist within the cell, and that can be taken advantage of using those ion-gated channels that we saw before. Inositol triphosphate, or IP3, is the last second messenger that we're going to look at. And what it is converted, so here's IP3, and IP3 is made by the action of an enzyme called phospholipase C, which converts PIP2 to IP3, okay, inositol triphosphate. And if we look in this overall grand scheme of a signal transduction pathway, we have all sorts of things we can take a look at. So again, we've got our G protein coupled receptor. It activates the G protein. Now in this case, our G protein is activating a phospholipase C. Phospholipase C converts PIP2 into IP3. That's our second messenger. IP3, and then we've got another thing we've seen before. We have a ligand-gated ion channel. In this case, an IP3-gated calcium channel. So IP3, our second messenger, binds to this calcium channel, to the gated calcium channel, opens up the gates, allows that calcium to flow from the high concentration of the endoplasmic reticulum into the low concentration of the cytoplasm, and there are all sorts of proteins that get activated once the concentration of calcium increases within the cytoplasm. Those proteins go on to give rise to the cellular responses. So this is a great example of a fairly complex signal transduction pathway, but of course they get much more complex than that. There will be entire classes that are dedicated to learning about signal transduction pathways that you will inevitably take when you transfer to wherever you might go. So these are some examples of signal transduction pathways, right? That is what links the receptor to the cellular response. And after you watch this a few times, make sure that you can describe the difference between kinases and phosphatases. Take a look at how kinases and phosphatases are involved in phosphorylation cascades. Define second messengers. Describe those second messengers' role in signal transduction pathways. And describe the function of adenylyl cyclase and phospholipase C.